Remember when we talked about the 4 macromolecules found in foods? They were proteins, carbohydrates, lipids, and nucleic acids. Well, today we're finally going to talk about those nucleic acids, which include DNA and RNA. You already know that DNA stores the information needed to make the entire organism. So whatever critter it is, the DNA contains the instructions to make that entire critter. DNA is in the shape of a double helix. It's a spiral staircase or a twisted ladder. Here are 2 models of DNA. You can see the double helix, the twisted ladder, and you can see the bases and how they match. Notice in this one, purple always goes with yellow. Purple, yellow, purple, yellow. All right. And red always goes with blue. Red, blue, blue, red, blue. All right. So that, let's say the purple and yellow represent A and T, and the blue and red represent G and C. And you'll notice that each nitrogen base has to have a complementary nitrogen base, kind of the equal and opposite base. The sugars and the phosphate molecules are part of the sides of the ladder. This is a more accurate model. You can see the shapes of all the parts more clearly. You'll see that these are the nitrogen bases. This is a sugar molecule, the little 5-sided piece, and this is a phosphate. So you see the sugars and the phosphates make the sides of the ladder, and the nitrogen bases make the rungs of the ladder. Once again, a blue and a red go together, so this is kind of like a C and G. Red and blue, blue and red, red and blue, red and blue. Then you can see that like the other one had yellow and purple, this one has yellow and green. So there's kind of like the A and T, yellow and green, yellow and green. They always go together. So hopefully this will help you understand DNA a little bit better. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. The building blocks of both DNA and RNA are nucleotides. Those are the monomers that make up the polymer DNA or RNA. They both contain a sugar. DNA contains deoxyribose, and RNA contains ribose. They both contain a nitrogen base and a phosphate group. The nitrogen bases in DNA are A, G, C, and T: adenine, guanine, cytosine, and thymine. The nitrogen bases in RNA are adenine, guanine, cytosine, and then it gets a little different. Uracil replaces thymine. DNA is only found inside the nucleus. It stays there because DNA is kind of like the original blueprints for making the cell. And you don't want to damage those blueprints. So they have to stay safely inside the nucleus where they can't be messed up as easily. Then RNA makes a copy of the DNA, plus this DNA blueprint is huge. If you unravel the DNA from one cell, it would be 6 feet long. That's incredible. So you don't need all the information just to make your eyes or to make your hair. And so a particular cell will only use the part of the DNA that it needs to make whatever it's making at the moment. What it does, it makes a temporary copy. You know, if you had this really old special document in the library, they wouldn't let you take that book out, but they would let you make a copy. So RNA is kind of like a photocopy, and you wouldn't copy the whole book, you'd copy the piece you needed. So the RNA makes a temporary copy of the DNA, And just like a photocopy, it's okay to throw it in the trash when you get done. It's okay if you spill your coffee on it, make a mess of it. It's no big deal because you're not going to keep it. So RNA is like a temporary photocopy. It makes the copy in the nucleus, then it leaves the nucleus and goes into the cytoplasm. Then it flows through the cytoplasm until it finds a ribosome. Ribosomes make proteins. RNA brings the instructions to make whatever protein the cell needs to make at that moment. Now, DNA is a double strand, a double helix, but RNA is a single strand. There's only one type of DNA for eukaryotic cells, but there are three types of RNA. They include mRNA, or messenger RNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA. The messenger RNA starts in the nucleus and comes out of the nucleus and goes to the ribosome. So that takes the message from the DNA to the ribosome. The transfer RNA are little transfer trucks. Okay, not really, but we can pretend. They're little transfer trucks that transfer the amino acids to the ribosome, and the amino acids are the monomers that make up proteins. So they're like the dump trucks bringing in the supplies. Then ribosomal RNA is part of the ribosome, and I really don't know how it works, but somehow it helps the ribosome know what to do and how to make the protein correctly. The whole point of DNA is that it stores information that codes for proteins, and the proteins make up the cell structures and run the cell's activity. Proteins are what make the cell go. Most of your body is made of proteins. When it needs to make a new part, it usually makes some more proteins. When it sends a chemical message that says, all right, now start doing this, that's often a protein. Yes, there are also lipids that can also send some messages, and remember, the cell membrane of the cell is a phospholipid. And of course, the carbohydrates provide the energy or the fuel for the cell, but most of your body is made of proteins. And proteins do most of the jobs or functions inside your cells. As we've already said, messenger RNA copies the DNA's information and carries it to the ribosome. Transfer RNA finds an amino acid and brings it to the ribosome to assemble a protein. And ribosomal RNA is already part of the ribosome and helps it make the new proteins. So how does a cell make these proteins? There are two steps, transcription and translation. In transcription, DNA makes RNA. In translation, RNA helps make proteins. So let's talk about those two steps. The first step, transcription, is when DNA makes RNA. What happens is, first of all, the DNA has to unzip so you can see the nitrogen bases. See, it's kind of like having to open a book so you can read the words. DNA is a double helix. There's two strands. So these two strands have to unzip and come apart so you can see the nitrogen bases inside. And that tells the cell how to make the proteins. The DNA is used to make a complementary strand of RNA. Complementary means kind of equal but opposite, and we'll learn more about that later. When the messenger RNA is complete, when it's finished making that, that leaves the nucleus through the pores of the nuclear membrane. Then the process of translation begins. You've got to translate the RNA into a whole new language, a protein. So after leaving the nucleus, the messenger RNA travels through the cytoplasm and docks at the ribosome. The messenger RNA has groups of 3 nucleotides, 3 pieces of those nitrogen bases, called codons. For example, you might have GAA, CCU, UAG. Those are all codons or codes found on the RNA. Each codon on the messenger RNA matches with a complementary anticodon. Once again, complementary, equal the opposite. That's why it's an anticodon. It's opposite. And it matches with a complementary anticodon on the tRNA. So kind of equal and opposite, and we'll learn more about that later. The messenger RNA waits at the ribosome, and the transfer RNA brings the amino acids to the ribosomes. Remember, amino acids are the building blocks for proteins. Each transfer RNA can only carry one certain amino acid. They can't carry any of them, just this one. So this little transfer RNA can carry leucine, and this transfer RNA can carry glycine. You get the idea. They can only carry one amino acid. There are actually 20 different types of amino acids that make up all the proteins in your body. So the amino acids are kind of like the alphabet, and the proteins it makes are kind of like the sentences. Then the mRNA codons match up to the tRNA anticodons. The messenger has the instructions, the transfer matches, and that puts the amino acids in the right order to make a protein or a polypeptide. A polypeptide is another word for a protein. Then the transfer RNA goes in search of another amino acid, brings it back, puts it into the protein, and continues on. And this whole process of transcription and translation is considered to be the central dogma of biology, the central teaching, the central belief. It says that DNA makes RNA and RNA makes proteins. And that is how everything works. So if you have a bacteria, the DNA makes RNA and the RNA makes the proteins, which makes the bacteria. If you have a dog, the cells in the dog, the DNA makes RNA, the RNA makes proteins, the proteins make up the dog. This is how every living thing works. So that's why it's called the central dogma, the central belief or idea.